developing these technologies using these new tools. The draft sequence of the human genome was, as you know, published in 2001. And after a decade, we have made a huge progress. Understanding the variation not only underlies the inherited susceptibility to disease
has left the conference
like cancer, but also using these technologies, looking at variation that happens de novo or spontaneously, and as well as somatic at the cell level. If you think about the human genome of 3 billion base pairs, only 1% of that encodes for genes. This so-called exomic region is important in the sense that any variation in these areas have a large effect size, whether the disease is inherited or happens at the somatic, at the cell level. So here, starting in 2008 and onwards, we have focused on these coding regions, and Yale was one of the first centers that has developed exome sequencing. And this, of course, is immediately applicable to discovering disease genes, which I'll give you in the next few minutes, some examples how we applied that, and reaching more and more complex genomic syndromes, such as de novo variation or somatic changes in cancer. The idea is very small, starting from the chromosomal DNA, sharing that. Initially, we used microchips. Now we're using solution capture to enrich for these coding regions, and then using DNA sequencing. As we develop this technology, of course, the immediate application for this is the inherited disorders, and specifically homozygous variation in the case of inbred families. And one of my main interests lies in how the brain develops and what are the genes important for that. And we have collected an extensive families from Turkey. And as you can see, this is a consanguineous union of first cousins. And looking at the MRIs, this is the patient's MRI versus a control MR. You can see that the brain is really small, is maldeveloped. And the challenge in these Mendelian disorders have been that these families are very small. There's early childhood mortality. And you really cannot use traditional genetic approaches. So by using exome sequencing, which was published in 2010, we have shown that a very different spectrum of brain malformations, ranging from these clefts that you see, brainstem atrophies, diffuse developmental abnormalities, were all due to this novel gene called WBR62. So this initially pointed out the robustness and the strength of this technology. Again, in this first applications of finding consanguineous or homozygous variation in inherited disorders. Just a few more examples. Occipital malformations. Here you can see smooth brain due to mutations in this gamma 3 chain of laminin. A recent finding from this month, which is, I think, is a fascinating syndrome where there's double cortex. You can see neurons are stuck here in the middle of the brain, but also neurons cannot stop in migration, and they're protruding from the brain, causing exencephaly. And this is actually also due to laminin mutations, but on a different chain in laminin B1. The most significant microcephaly that I have seen from Eastern Turkey, normal brain, age match to this brain. Hard to imagine that this kid is still alive. Was the only affected in the family, was the only one in the world, but by using these next generation sequencing, we were able to show the mutations in this gene called ND1. And since then, again, another interesting syndrome in a disease, in a gene called UCHL1, which also have implications for cancer. This is the most abundant protein in the brain. It's a ubiquitin ligase. You can see the patients have these significant ataxias due to cerebral atrophies, optic atrophy, and in their 30s and 40s develop this brain atrophy. And again, this technology was very robust in finding these genes in Mendelian disorders. For the purpose of this morning's talk, I'm not going to go into the use of these also to understand the complex genetic architecture of common diseases. In my lab, it's specifically focused on intracranial aneurysms, but by using these technologies, we also are starting to shed light into how these aneurysms form. But in regards to the tumors, I think similar to the other interests in my lab, the overarching goal, aim in my lab is to understand understand the biology of different cancer, and we focus on brain tumors in the lab by using these next generation genomic technologies and use that finally to bring this to clinical medicine. And I'm happy to report the first DNA that I think we collected in the lab was 1993. After nearly 2 decades, we are at that point that we are discussing daily on how to use these technologies to change the care of our patients in the clinic. So, of course, the first thing we have to do is to understand the genomic variation and understand and catalog these. And we're lucky in the sense that it turns out that the way a cell can create cancer is limited in number. It's not infinite. So I think we will be able to obtain a catalog of these by using this subclassify tumors, which are easier for some benign ones like meningiomas, as we will discuss. A lot more difficult for as it becomes more malignant, and as you know, the most malignant brain tumor being the glioblastoma multiforme, GBM, and gets very difficult because not only one GBM is different than the other one, but I think the genetic heterogeneity within a cancer is also very important in designing these therapies, and this has to be taken into account. By using these technologies now we can come up with a temporal history of a cancer, and we can understand which mutation has happened when in the evolution of this cancer, and I think this will have implications to these individualized treatments. So if I think now, as opposed to a couple of years ago, about variation in cancer, I use the simplest example of a homozygous mutation leading to an extreme brain phenotype. But if I look at a GBM sample now, I think about the following. I think about the coding mutations. I look at the total number. I look at the protein altering ones. I look at structural variation, not only small indels that affect these protein coding regions, but also large copy number changes. We look at translocations, we look at fusion events, and we consider gene expression not only at the microarray level or RNA sequencing level, but also looking at transcriptionally active sites or transcriptionally silent sites, such as with the methylation analysis. We look at the mutational signature. Is there transitions? Is there transversions? What are these dominant mutations that underlie a cancer? And taking all of those and applying them and understanding subpopulations within a cancer like GBM, and understanding, again, driver mutations and copy number changes for each of these subpopulations. Looking at the clinical data, learning from outliers, the long-term survivors, and ultimately coming up with the targets that are defined based on all of these analyses. And as you start, hopefully, which will be starting soon here, designing these targeted therapies in combination with other therapies to define their response to therapy. This is a very dynamic environment, similar to evolution. With selective pressures, they respond differently. And as we treat these with either chemo or radiation or surgery, we are selecting and taking sides of one subpopulation over other. And I think that's really important to take into consideration on top of the natural mutation that happens in these cancers as they grow. So what we have started doing in the last nearly 3 years is starting with the surgery, obtaining the tissue, and using different approaches, as I tried to summarize, looking at all these different levels. For example, combining exome sequencing with methylation and RNA sequencing, or in few cases, which I think this is going to be very important, to use whole genome sequencing, especially in difficult tumors, and in understanding response to therapy. We also culture pretty much every brain tumor that is resected here, followed by in vitro chemical profiling, but also understanding if there's targeted therapies, whether we can choose drugs based on this in vitro modeling, which is quite challenging because, again, when you, I won't go into details of that today, but as you select and culture a subpopulation from a cancer, you really are selecting a specific subpopulation, which undergoes, of course, changes in vitro. So that's not very straightforward. If you think about brain tumors with their incidence in age, the really top ones are gliomas, meningiomas, along with acoustic neuromas and pituitary tumors. I will mainly, as I mentioned, focus on our work on meningiomas and gliomas, which are the most common benign and malignant brain tumors. When you think about meningiomas, these are the tumors that originate from the from the meninges that surround the brain. They are pretty benign histology. Over 90% of these are histologically benign. But the origin of these tumors inside the brain, for example, the olfactory groove ones, might make the clinical course difficult in the sense that they tend to invade structures such as optic nerve or encased carotids, leading to significant neurological deficits. Looking at the MRIs, they diffusely enhance. Here's an olfactory groove MR in the middle of the anterior skull base, again, invading both optic canals, leading to blindness versus a convexity meningioma with diffuse enhancement, but some malignant degeneration. As we discussed, the treatment is mainly surgery, which are pretty straightforward in easier locations, but they typically, they could recur, especially in challenging surgical areas such as skull base, radiation used for residual lesions, or if they come back, but there were no effective medical or chemotherapeutic treatment options. So in the lab, Victoria and Zeynep took 50 of these tumors, and they have done exome sequencing and looking at the results. Consistent with their benign histology, the number of protein-altering mutations were really small. It's less than 10. And taking these and combining the data and doing a permutation analysis to identify the genes that were mutated more than just expected by chance, they have identified these 5 genes. The top gene were NF2, which we're all happy because this is previously reported that NF2 mutations are identified in 50% of sporadic meningiomas, but the next few of them were quite surprising. So if you consider these genes, the first one that is mutated in almost half of non-NF2 meningiomas is a molecule called TRAF7. Not much information in the literature. It's a ubiquitin ligase, has these so-called WD40 repeats, which are important for protein-protein interaction. And what's known in the literature is that it can signal through NF-kappa B or through junk signaling. And most of the mutations, as seen here, that we have identified affect the WD40 repeats, suggesting that it affects one of these pathways. I won't go into details of those studies, but we have done quite a bit, in vitro at least, studies looking at that. It appears that NF-kappa B is activated, signaling is activated in these meningiomas. Next one was this molecule, KLF4, that most of you might know, especially those of you who work with stem cells. It actually won the Nobel Prize for one of the molecules that won the Nobel Prize for Yamanaka last year. As you know, these 4 factors that Yamanaka has identified are used for producing pluripotent stem cells, starting from terminally differentiated cells such as fibroblasts. KLF4 and other 3 molecules can lead to formation of stem cells, which is a very, very powerful technique. We have identified a single recurrent mutation in multiple different tumors, this K409Q mutation, which is in the DNA binding domain of the molecule, suggesting that it changes the specificity of downstream signaling for KLF4. And the hypothesis there, of course, is that this mutation leads to a less differentiated state, more stem cell-like state for the meninges, and that's important for tumor formation. Next one was, again, a recurrent mutation in AKT1, E17-cain. This is a known mutation that is shown to activate PI3 kinase signaling, has been identified in multiple cancer. This is a schema from Cosmic. You can see that there's a huge peak in the 17 amino acid, and it's one of the ways to activate PI3 kinase and survival signaling. And finally, we have identified activating smoothen mutations, which you might know is involved in hedgehog signaling, which is important also during embryonic development. And it is the activating molecule for hedgehog signaling through Glial transcription factors. And the mutations in this pathway, which will become relevant later on, have been identified in similar brain malformations like the one that I have shown you, leading to holoprosencephaly, where the brain in the anterior midline fails to split into 2. And as I said, that will become important later on. So based on this initial information, Victoria went back and did targeted sequencing of 250 more meningiomas and identified mutations in NF2 and other molecules. And actually, this led to some interesting results because of the mutual co-exclusivity of these mutations. For example, if you look at the NF2 tumors, they almost never overlap with TRAF7 mutations, and in contrast, TRAF7 mutations almost always coexist with the identical mutation in KLF4 or AKT, so these are coexisting, and small mutations, hedgehog signaling, defines a third group. Besides the somatic mutations, the surprising results also came from looking at the copy number variation. And these are circus plots. Every circle represents a different tumor. Here the amplifications are shown in red and the deletions are shown in blue. You can see SMO and TRAF7 mutations have no copy number variation, whereas all NF2 tumors, also can be seen here, has chromosome 22 loss. And as you go from benign to more atypical tumors, the number of genomic changes, increase. So this suggests that these are really biologically distinct classes, which also can be seen in gene expression. You can see, for example, NF2 tumors versus non-NF2, and looking at gene expression patterns, Akdes and Zeynep have shown that they also have activation of different pathways. So overall, this summarizes the at least 85% of meningiomas can be classified in three different groups. Those due to NF2 loss and chromosome 22 with chromosomal instability, leading to formation of meningiomas, TRAF7 in junction with AKT1 or KLF4, or against mutant, and we're working on the other, and there are really interesting findings on the remaining meningiomas too. So I think we're up to 92, 93% now. So of course this is all good, looking at the mutational, but does this have any clinical relevance? So to answer that question, we first looked at histology, and the answer was yes. There are 15 different histologies that are described for meningiomas, and the correlation is perfect for some of them. For example, this one that is secretory meningioma that is characterized by these periodic acid shift positive collections. Clinical relevance being that these are the ones that lead to the most amount of brain edema. And not surprisingly, looking at the subgroup, they have increased expression of EEGF. But we had 12 of these tumors, and when we looked at them, they were all TRAF7 KLF4 mutant. So it correlated with histology, but I think the most fascinating thing was, now looking at the mutation profile, we went back and reviewed the MRIs, and looking at the origin of these tumors within the brain, we can actually tell the mutation profile. There are significant gradients. If you look at, for example, the skull base, all NF2 mutant tumors are located posteriorly and laterally, whereas the non-NF2 tumors are in the anterior skull base. And you will remember that I mentioned the holoprosencephalus with hedgehog signaling being an anterior midline defect. All mutant, so hedgehog mutant tumors were located in the anterior midline. Similar correlation existed for the convexities, where the NF2 mutant tumors were localized to the posterior, whereas none of the NF2 tumors were anteriorly. And this appeared to correlate with the embryonic origin of the meninges, even though it's poorly understood in humans. This is, as you can see, from a rodent. So based on these, of course, now understanding the majority of the genetic base of the majority of the meningiomas, we went into whether we can come up with some new therapies, especially targeted therapies. And as I mentioned, we culture all these tumor cells. Katie, looking at arachnoid cells where these tumors originate from versus other mutation phenotypes, has seen that there's different morphology, their growth rates, and as I mentioned, gene expression profiles are different. I won't go into the details of that, but we have done in the West Campus a chemical screen, especially for TRAF7 and mutant tumors, coming up with a lot of hits that she's doing now, the secondary screening. So overall, I hope I was able to convince you that we were able to explain majority of the meningiomas with mutual exclusive distribution of these mutations in these different tumors. They have distinct potential for chromosomal instability, which underlies the potential for becoming malignant. And you typically see those, not surprising, with NF2 mutant tumors in the convexities or in the posterior fossa, whereas the anterior skull-based ones almost never become malignant. We correlated those with histological appearance. I didn't go into details, but also as well as with gene expression and acetylation based on chromatin immunoprecipitation sequencing. So now looking at the origin of these tumors and thinking that the activated mutant mutations as well as hedgehog signaling has been implicated in other cancers. And there's an FDA-approved drug for metastatic basal cell skin cancer, which hedgehog signaling is seen. you can imagine that for these difficult anterior skull-based ones, we can potentially use hedgehog signaling inhibitors, and we are testing that in the lab. Of course, there are AKT1 inhibitors too. It's harder to judge whether they will be effective because they typically coexist, as I've shown, with TRAF7 mutations. But for TRAF7, we have, as I've shown briefly, good hits that we think will be relevant if we can show their effectiveness in vitro. So this is, again, taking one level higher. These are benign. But the difficult ones are these malignant ones that are glial tumors that, as you know, from a precursor, glial precursor, you can get oligodendrocytes or astrocytes leading to astrocytomas, oligodendrogliomas, which could be benign tumors, and then going to glioblastoma, which is a completely different landscape. So to be able to understand that, we have done genomic sequencing of GBMs, and the results are summarized of all those 3 billion letters here in the circus plot. The most outer circle shows the coding mutations going to the copy number variations. You can see, for example, chromosome 10 loss here, with all calls being homozygous. And these gray spikes show the translocations within the chromosome and translocations across chromosomes. So this is a very different landscape than benign meningiomas, and I think it will require a lot more work, which has been already done through the work TCGA, but I think we have additional information to add. Just looking again, the low-grade ones first, Victoria was able to classify them based on their molecular signature. So the cell of origin, whether they're astrocytes or oligodendrocytes, they appear to have mutually exclusive mutations. For example, although most astrocytomas have P53, usually by allelic loss, they also have ATRX. Oligodendrogliomas are characterized by 1p19q deletion, and the mixed ones are mixed signature. And this was already known in the literature. So now we have done over 222 exomes and 52 whole genomes, and we are working on the molecular subclassification. And as mentioned before, looking at, for example, 2 GBMs, really they are very, very different among each other. I won't go into much details, but I have mentioned that looking at mutational signatures, for example, transitions versus transversions, also is able to classify these tumors. As you might know, there are 2 major pathways to form glioblastomas, which is either a transformation from a low-grade glioma, so-called secondary glioblastoma, or primary glioblastoma without evolution from a low-grade. And the low-grade ones are characterized typically by IDH1 mutant, which are shown in red here. And you can see that overall, even though it's not perfect, the mutational signature, looking at transversions and transversions, actually have an effect on this. And I think this will be relevant. As relevant is the number of mutations. So if you look at the number of mutations here versus the fraction of copy-altered genome, you can see that GBMs are very different. And Tom was very excited about this, I think, for now has been shown for colon cancer and for endometrial carcinoma. There are hypermutated phenotypes, typically due to polymerase epsilon mutation. And these patients appear to have the best prognosis. And this is now true for GBMs. So overall, in order to use any of these targeted therapies, I think we really need a good molecular understanding of each of these cancers, given their genetic heterogeneity and adaptive responses. I think we have a lot to learn from our failures. In collaboration with Roy Herbst and Tom Lynch, what we're doing now is to looking at the TCGA data, which Chris Sanders from Memorial Sloan Kettering was kind enough to share with us, to looking at these and coming up with this basically a pretty straightforward software where exome data is loaded, which then leads to identification of these genes and mutations that are actionable. We're now running this program on the TCGA data with lung especially to see what percentage of lung tumors will be actionable if we started a clinical trial today. With GBM, it's more straightforward because the typical loss of chromosome 10 with P10 loss, activation of PI3 kinase pathway, and EGFR amplification accounts for 80 to 90% of GBMs. So you can imagine that that's more homogeneous. By using this program, you can choose the tissue that is of interest to you, and then be able to identify the cosmic entries that have been identified, for example, in IDH1. That you can see, not surprisingly, multiple tissue types with multiple mutations have been identified, and it also generates a more detailed Excel sheet if you really want to go into more detail. So in conclusion, I think that these future generation technologies, as we learn them and as we learn how to apply them to more complex scenarios like cancer, now allows to this molecular subclassification in between tumors, but also within tumors to apply these subpopulations, which appear to be clinically relevant. For example, there's a good paper on breast cancer which shows that the portion that metastasis is a very small portion of the all breast cancer tumors, like less than 5%, but it's always the same molecular signature that metastasizes. So of course, we're hoping that this will form the basis of individualized therapies, which in my mind will include the targeted therapies that we've discussed, but of course has to be combined with what I call as more non-specific therapies, such as immune, especially active immune therapies and anti-angiogenic, and hopefully others. So I think I'll stop there. I won't acknowledge all the people, but try to show their pictures, and in a few minutes, I'd like to take questions. Thank you.
Sure.